business of breaking things down bit by bit so we can put the bigger picture back together. And that's what I tried to do in this series of lectures. There are key events in we're going to look at life on Earth. We're going to look at the beginning of time on Earth, and there are certainly different viewpoints on all this, as there is in any history book, because no one was really there to record this, so a lot of it has to be put together, and it can be controversial. Some people say, well, it's all part of a great design. Well, maybe, I don't know about that. In science we try to explain things, so we're going with what's explained. And even then, it's theory. People put it together based on logic, and parts of it will make sense to us, and part of it will be like, I don't know. And that's kind of the basis of our discussion. Well, let's find out, what people know and so forth when we launch that discussion. In this, then, what I'm going to do, and you'll have the slides also, but if you want to put a timeline across your notes and feed events into it, that might be a nice way to study or think about how to put things together when you're getting ready for your exam prep. I'll try to hit major events. I'm not going to micromanage it for you. I mean, after all, it is 4.6 billion years. A lot has happened. But I like timelines because they help us to organize, right? So that's what's going to happen. We'll be going through there, and if I have this right, different events will kind of light up as we go and kind of be a traveling bookmarker for us. So here we go. Our timeline really has 4 big points to it. Each one being 1 billion years plus a little bit more, and so somewhere in there, Earth was formed. But you have to think about what Earth was like. Really a little free water on it, and one of the reasons is Earth is really, really hot. Okay, so that's how things are. There's no oxygen, that kind of thing. And we gotta start building stuff up from there. You probably had some of this in general biology, but we'll start putting the microbes into it more. The first part that we start looking at that sets the stage for life is making chemicals that are usable. Now, inorganic chemicals are on the menu for microbes. That's a little different than for us. We need organic molecules, as you all know. And we want to talk a little bit about chemical evolution. And I just want to say before I get into that, this isn't the same kind of evolution that we talked about on Tuesday, where we were saying, well, Darwinian evolution has to do with selection, and as the narrator was saying, we have genetic mutations and some kind of things tying together into continuity. Chemical evolution is more the idea of one type of thing giving rise to the next, but there's not that inheritance kind of factor in it. So in a way, it's a great term. In another way, it could be confusing, like when we say reproduction with viruses, we want to be, kind of have a good idea of what we're trying to talk about. The chemicals that exist are inorganic. So we see water, nitrogen gas. Methane is doing really well on early Earth, as is CO2. Iron in different factories. Most of these, awful lot of reduced things because we really don't have a lot of air, oxygen floating around as we know it. The advent of organic chemistry is thought of to perhaps have had its start through lightning strikes to the Earth, or basically the input of harsh energy. It could also have been from high radiation coming from the sun. Realize early Earth had no ozone, had no nice atmosphere buffering the Earth. So a little bit of openness on that. The Urey-Miller experiment, contentious as it is still worth considering. What these fellows did is said, well, let's set up conditions that we think early Earth was like. Let's set up some water with the types and gases with the types of chemicals that we think was the atmosphere and water in early Earth, and then let's see what we get from this. Okay, And the idea is taking ocean water, having some of the gases we just saw in the last slide, and then bringing it in, heating this. The Earth is hot, so you're getting vapor material happening, so they get the reaction temperature up. And then they had a different reaction chamber over here where they have electrodes simulating lightning. And then a condenser, much as you've probably seen in any chemistry class, condensing it, and then refluxing it. Doing this over and over again. Because remember, early Earth, nothing but time, okay? They have no early Earth, nowhere to go and all day to do it, right? So, I mean, it's just continuing to reflux. And from this, they got interesting materials. They started to get some of the early biomolecules, amino acids, nucleic acids, monosaccharides. And so the idea is, well, did that really happen? No. Is the Miller-Urey experiment criticized? Oh, yes. You know, it could have just been something completely different. Maybe a meteorite hit and a whole bunch of stuff came in. We can go and look. That'll be kind of part of our little exploration. But the point was, at some point, you had to start getting monomers, and that did seem possible as by simulation of what early Earth conditions were like. Well, organic molecules are a good first start, but that doesn't really go for life. Why? Because as cool as amino acids and nucleic acids are, they're not going to make life. You need polymers, and you need something to make energy, break stuff up, and tear stuff down. And so there's no enzymes. So different theories on where these may come from. One is that there's really high electrostatic forces that do exist. If you've never, as a biologist, contemplated a clay particle, you should for a moment. The size, when you pick up soil, I don't know, maybe in ecology you still do this. I took a few soil science classes, so we had to feel soil. Okay, well, you know, soil is all a mixture of particles. Okay, the big particles, gravel, pebbles, okay, you kind of see those, you know, shoot them with your slingshot, whatever you want to do with those. That's really easy. When you pick up soil like in a farm field or a forest, you can feel it. What you feel in your fingers, down to what the smallest thing is you can feel sand very easily. You can still feel, to some extent, silt. You cannot feel or roll as a single particle in your hand clay. Clay is too small because clay is about the size of a bacterium. You know, it's a few microns in size. Okay, now, one way you can get a feeling for clay in any soil is to get your soil sample, mix it up, and just pour the slurry into like a graduated cylinder. Maybe you do this in one of your classes. One way we would separate out the percent of different particles is to do that, and gravity, like a very slow centrifuge, which gravity is, will pull things down by their size. The sand and the bigger stuff all fall out right away. The silt, and you can look up on tables, the silt takes, you know, an hour or two, and it's coming down in this layer. Clay, you come back the next day, and it's still kind of settling out. You get an idea, wow, this is small stuff. Now, why do I care about clay? Well, I care about clay quite a bit in this context, because when you have, now you all have talked about the surface area to volume ratio when we start getting to smaller things. We're talking about mainly living things. Inorganic things are still really important. Clay particles have an incredible amount of surface area, and depending upon their composition, can really affect the locality of things that coat them. Okay, the clay in this area is made up of mainly what? Morillite. If you took soil here and you put it in an oven, okay, and you brought back, let's say you're making a pot, a clay pot, what color would it come out after you fired it, do you think? Come out looking like dirt? it comes up bright red, okay? What does that tell you? Anything? What would give soil a red color after heating, or really, in a way, really oxidize, pushing it over to an oxidated state? Iron. Yeah, we're really high in iron, okay? And that's not unusual. You go to the, you travel through the American Southwest or West, the native people talked about the good red earth. Not uncommon to hear about that in places like Vietnam and Cambodia. Iron is a big deal. Now, I'm not saying iron's the only thing, but you start getting a high surface area with some kind of electrostatic interaction. Now, it could be the type of thing that could bring monomers close enough together to start making polymers. It is a theory. Okay. There's other theories on the books. But I wanted to put them on passing. Clay can also be very layered. So you can start putting things in between layers, and you can start messing with molecular things, molecular bonds and so forth. So it could be that clay led to some of the first early polymers. Okay. Things that also have started to have to happen then is a buildup of lipids, a synthesis of phosphate, a membrane vesicle that enclose the cell's biochemical machinery. We've already talked about the importance of the cell membrane. Whether the first membrane was a phospholipid or not known. Phosphorus was a good one to go to. I mean, the other thing would have been to make the membrane a sulfolipid. Okay, but phosphorus is a little bit more stable. It has less oxidation states. So it did seem to make sense. But even if you had a polymer of lipid, you could start thinking about making vesicles. Okay, maybe in cell biology, I don't know what they do anymore. We used to take cells and we would blow the insides out of them, and we'd have these things called liposomes. Basically, it'd be a cell with a membrane with no cell in it, and then we would put in certain reactions that we'd allow to react in that type of little vesicle to study things very specifically, or to look at the characteristics of membranes. Don't know how cell is done these days. We were very experimental when we did, but we had just gotten off the Mayflower, and someone gave us fire, and then we started doing cell biology, so all that kind of stuff was happening. So some other ideas then, in the literature, it's not uncommon to see, there's Scientific American. Anyone read Scientific American at all, ever? It's at your newsstand. It's a great thing if you're a scientist and you like finding what's going on. What's nice about Scientific American is they invite articles and they cover things. So you're kind of getting a journalistic point of view, but they definitely keep it very factual. I've did with friends. Some of my friends studied spiders and the biomechanics of webs, and they came and interviewed those guys when I was at Vancouver, and they're very good at what they do. Anyway, it's a good source. It's not exactly peer reviewed, but it's really high up there and it's popular. You can grab it. It's probably online, I'm sure everything is. Anyway, they're talking about vesicles and this whole idea of clay starting to help first cells. So there's evidence that's out there, and here's the idea that you could put in a simple fatty acid. This one's not even a phospholipid, and by self-assembly, you can get a vesicle. You probably know this from perhaps chemistry, inorganic chemistry. You put on. Do you do a critical micelle concentration? Does that ring a bell at all? CMCs? Well, maybe write it down and look it up, or I'll tell you either way. But CMC, every detergent has a CMC characteristic, and that is and the way to think of that possibly is that if you put a drop of soap onto, let's say, a beaker of water, you can add a concentration that's low enough where it'll just first coat the top of the water. Why? Because the lipid wants to get away from the water and it does a single coating. Okay, you go past the CMC, and then it starts to want to double up and eventually make vesicles, make micelles. You probably remember that term from chemistry. I hope, otherwise write that one down. But micelles then, critical micelle concentration then, is essentially a lipid bubble, if you will. That's usually dictated by the size of the fatty acid and how many carbons you have. Okay. If you're doing a lot of work with dissolution or work with membranes and different detergents, you would see this in the literature quite a bit. Okay. So my point is you don't, at this point, if lipids were around, you might not have to have special enzymes just to make the first vesicles, just to make the first micelles. You know, and micelles can, under certain circumstances, become double layered, but even if they're single layered, now you've established outside and inside. Now, are these micelles fragile as can be? Oh yeah. You know, they don't have the cell wall. That's not going on yet. You know, could they be blown apart? Yes. And all those things that you know about are still on the table. It's not like that's gone away. On the other hand, you know, there are places, and you've been places on Earth, where the wind doesn't even blow. There could be some very different environments where you might have this type of thing go on. Oh yeah, this is myristic acid, but it doesn't matter. So you can get these microspheres that start to form by self-assembly. Okay. The putting together of DNA and RNAs is a little bit harder, and I go back to one person, this book, I think I have a slide of it next, on microlife. The questions of how did life evolve on Earth is really a good thing. We used to read it in class, but it's getting so old now that I felt that maybe, you know, time could be spent on other things. But what Margulis' theory is that you can take cyanide, basically, and if you start looking at the nucleic acids, you can start seeing that you have different assembly of cyanide molecules, and you can get the nucleic acids, or the nucleic bases. Now, at first you go like, oh, okay, well, you know, I'll buy that, or I don't buy it, whatever, but I thought it was pretty clever to think, well, what would be on Earth? Well, cyanide was pretty prevalent, you know, I mean, it's not that hard of a molecule to have with the compounds there. And to think about, and you know, if you start really, if you want to go and look at her actual treatise on this, you can start kind of tearing the nucleic acids around, and you see that pattern, you go like, well, you know, she's a very creative person, you know, is it just her imagination, or is that how it works? Don't know, it could be something we could look into a little more, but I've always liked the cyanide theory for the nucleic acids, okay. When we think about the origin of life on Earth, there's several theories. The surface origin hypothesis contends that the membrane-enclosed self-replicating cells arose out of the primordial soup, rich in organic and inorganic. So we've kind of covered, you know, at least an idea that gave rise to that, with different temperature fluctuations and mixing of other things, meteorite impacts, dust, so forth. It seems that there could be reasons that work against it. The inoculation theory is neat. And I, you know, as a scientist and a microbial ecologist, I can't say that's not how, you know, that's not how it works. You know, it's possible that a meteorite, microbes that evolved somewhere else in outer space, why couldn't they hit the primordial soup at an opportune time and start growing here? Certainly a lot of ideas about diversity and so forth might be solved right there, but still begs the question. So while I like the idea that we were inoculated, and there's a few other theories out there, I stick to what do we know about Earth? That's the thing we know about the most. Will we discover meteorites with truly real DNA in them? The microbial fossils or remnants that we think we see in meteorites, I think they're highly contentious. But you know, read the literature and see what you think on some of that. The subsurface origin is really interesting. I like it. It states that life originated at hydrothermal springs on the ocean floor. Do these still exist? Yes. So possible that life originated there. Some of those original life forms are there, so they send down things to go check these things out. Conditions would have been more stable than at the surface. Okay, you have UV light not being impeded at all with any ozone or anything. You have windstorms and other things happening. Maybe it'd be better that this occurred if a steady state and abundant energy. Okay, we have hydrothermal energy going on, and we have different raw materials that could go into some of the first metabolism, which we'll look at in a moment. So the other nice thing about the hydrothermal vent idea is that you also get a gradient. Okay, as these gases and things move in and start mixing with ocean water, you also have a cooling effect going on. And even though this might be too hot to get any kind of cell going, you could have like this refluxing and condensation type of thing going on as you move up into the hydrothermal vent. Okay. For enzymes, like I said, we had an idea that inorganic particles might have some kind of action that could be it. The other thing that shows up in the literature are ribozymes. Did they talk about ribozymes? Anyone take biochemistry? Did they talk about ribozymes? Okay. How extensive is it? Just in passing, kind of?
Covered it briefly.
Yeah. Well, the upshot is that, when we're in general biology, you say enzymes are proteins. Well, and most are, that's fine. It's also possible that if you've been able to make nucleotide bases, so you need a sugar, a phosphate, which is, around, and then you have cyanide that you made into bases, and now you can make polymers from that you could then start using an RNA-based polymer to catalyze reactions. So that's one of the ideas. Here's a good book, I think, to read if you like this kind of stuff. Microcosmos. Okay, Margulis, and he's probably, I've said was married to Carl Sagan, which a lot of us growing up back then, Carl Sagan was like the science guy, on the Discovery Channel or something. He told us about all these crazy things, and so Lynn and Dorian decided her son started to write books. I think Lynn and Carl tore the sheets at some point and didn't think it was a good idea to stay married. But this talks about a lot of these different ideas in depth, and you get it from a person who has thought very deeply on it. You probably, this is an older book, 96, you can probably get it for a penny on eBay or something. I would say it's worth the read. We've read it in Advanced Micro, and the students go everything from, wow, that's really cool, to, oh, I don't believe any of it. So, I mean, I'm not here to make anyone believe anything. We're here just to discuss ideas, so it's kind of fun. Well, right about 3.8, 2.9 billion years ago, we start thinking about that probably the first organism could probably be formed. Conditions could have been right. First self-replicating organism. Take a little closer look at that. We have self-replication because we have some self-assembly going on amongst the molecules. And we also then realized that early life on Earth was, I would say almost definitely would have to be RNA type of life. Okay. RNA with ATP as a possible energy currency, that could start happening. RNA was just a smaller molecule that was able to be more readily synthesized. The question that usually comes up among students is, well, why did we ever move to a DNA-based life form? I understand viruses might have it, but you heard Tom Schmidt, viruses, they're kind of over there, right? So let's just stick to what we consider to be, we know is life. Why move to DNA over RNA? Okay, let's just assume first life was RNA. Let's just stick with it. Why mess with DNA? Any ideas? Maybe you've covered this. Any you've talked about in other classes, or thinking about? Yeah.
DNA is more stable.
It's more stable. Why? What makes DNA more stable? I think you're right.
There's 2 strands.
2 strands, well, you know, RNA, like, let's look at tRNA, it can be double-stranded in places. Go ahead, Jake.
The bonds that form the helix, the hydrogen bonds within it that allow it to have this spiral shape and then it becomes more compressible as well.
Yeah, the hydrogen bonding in double-stranded RNA is still the same hydrogen bond, but combined with that idea of that double helix that stays double, you know, into a double helix all the way along, it's just thinking that little shift in the bonding of nucleic acids does make it more stable, okay? Therefore, what probably moved things towards RNA life is that the selection against RNA was so high that it was not keeping a good, you know, it wasn't being good about keeping the information from generation to generation. Of course, if you're not doing a good job of that, you're being selected against. But yeah, DNA in that double helix, aside from being, you know, that real pretty, kind of beautiful kind of, you know, double spiral that we all, you know, enjoy artistically, also has an inherent stability to it. Yeah. Yeah.
What at the beginning would have driven life to reproduce?
Thermodynamics.
Because it takes a lot of energy to reproduce.
Why would the,
Why would the first self-replicating organism have come about? What would have caused it to want to pass its gene off to divide or whatever? Are there theories?
oh, so
about that? Yeah.
what is life? Yeah, they don't,
Why is reproduction a
Yeah.
thing?
we don't know.
Okay, that's my question.
Does anyone know? I don't know. Great book, What is Life? Yeah, I mean, I like the idea, and I don't think we know why life decided to replicate. Okay, we find that it became energetically favorable, you know, and maybe that in itself just allowed this series of reactions, but yeah, why bother? Why not just make more polymers? Why not just, you know, fill the earth up with, you know, other stuff? You know, we know why life continues to go, because selection for those reactions are favored, and therefore it procreates. Okay. It takes a lot of glamour out of, you know, Valentine's Day when you talk about life that way, but I think life does, for me, it boils down to the question of thermodynamics and and probability. But more than that, you know, I'm not sure. Last, you'll see this sometimes, Luca, okay? The last universal common ancestor. This is that, you know, when you see the base of the tree and you see 2 things coming off, you know, it's usually the bacteria and the archaea, and then the archaea and the eukarya come off right after that. That base of the tree, that last universal ancestor, is theoretical. You know, there's no hall, there's no little vial in the Museum of Natural History that says, here's Luca. Okay, and probably never will be. It is a theoretical place, but it works. You see how they carve away at getting to the theory. Okay, Schmidt was talking about, Karen Nelson was talking about, you know, Thermatoga and, you know, what happened when they started sequencing. Like, holy smokes, it seems like it's really close to there. You know, could there be a little blip of this someplace? Maybe theoretically. I mean, if it really did, you know, evolve in a hydrothermal vent, I mean, yeah, maybe someday someone will put a probe down there and find something. It's just, that's it. That's the one that has both of them. You know, it's possible. I don't know. No one's found it yet. So, in the RNA world, okay, you start out with, you know, sterile Earth, this prebiotic synthesis. We have RNA here shown with a hairpin loop on it and some double stranding, which I wanted to reinforce. It probably was that. You start getting more self-replication in there, and where this finds a home to do this inside of either a lipid or a lipid protein vesicle that keeps, again, outside from inside. Okay, replication, could occur in this level outside of a vesicle, but it's gonna be lost, it's not gonna be carried on at all. So it's possible that at least the RNA part of this thing could have started on that level. Where things get to be more interesting then is, well, we've talked about this, at some point you start moving into the idea of a DNA-based cell over RNA through stability and again thermodynamics. This is just taking that idea of the thermal vent and moving it more towards what might be going on. Basically, they're saying that you have the monomers, polymers, and an area that is close enough where you can have these reactions localized. Okay, one thing I don't think you'll ever read in the book, but I hope to leave with you is one of the things that goes against life and against feeding and trophic nature is diffusion. Okay, also went through, we had to overcome the idea of diffusion in a big way to get onto land. Okay, as aqueous as you are, you're still in kind of a semi-permeable sac that keeps you from diffusing away. So having molecules, having materials that possibly could be made life and floating freely in the ocean bothered a lot of people. And they started saying, well, but you could have something like this going on. The evidence to tear apart a thermal vent or some of this material, I'm not sure is well thought of. Talked about DNA already, so I think we can go past that. And pretty much been through all of this material. So if we have the nucleic acid and possibly have self-replicating information centers, we still need now to get energy into the cell, to get early metabolism going on, to have any kind of meaningful cellular organization going on. So in here, probably the first type of chemical reaction that microbes or pre-microbes ever had was going to be chemolithotrophic. Okay, and you don't see this type of reaction exactly in the Winogradsky, but you do see the movement of reduced sulfur to something more oxidized and oxygenic kind of reactions going on. So if we look at early metabolism, anaerobic and likely chemolithotrophic, obtain carbon from CO2, so autotroph for the carbon source. Here we have hydrogen gas, carbon dioxide, yielding water, okay, and then some type of carbohydrate, all right? Contain energy from hydrogen available in the environment or from other sources, generate energy reacting with hydrogen sulfide or UV light. You could also generate some kind of energy source by just basically letting light as an energy create, move protons around. So here we have FES going into FES2. As far as I can figure out, it's close to being like pyrite. Perhaps the chemist can correct me on that. I'm not sure that it's exactly pyrite. I've talked to Dungy about it a few times, but I'm pretty sure we're close to it being like pyrite. So we have our proton, basically our electrons, being going to an electron acceptor and therefore generating hydrogen sulfide and that type of pyrite. So from here, the hydrogen sulfide can also then serve as picking up, being more, being regenerated from sulfates, if you have that going on. And then they're also thinking at some point you have to have some kind of enzyme that's moving hydrogen around, which would have to be a primitive hydrogenase. And again, this would have to be a membrane. It has to be occurring on that level. You don't see a cell wall. You don't have localization of protons and movement of electrons. It's going on, probably terribly inefficiently. at this point, but, you are, probably in a sea of this stuff, so it'll work out that way. And you can get the ATP if we have an ATPase. All right, early forms of chemolithotropic metabolism would have supported production of large amounts of organic compounds. So once you could start getting this machinery going, you could start producing a whole bunch of other types of compounds, and then life can get more complex. So probably not altogether known, but getting into this idea of the two domains of life, that's probably happening very shortly thereafter. Start to see RNA polymerase giving rise to DNA polymerase. There's probably still organisms living as hypothermophiles. Most of the Earth is still that way. And probably not really setting up the eukaryotes yet, but that's probably going to happen a few, part of a billion years later. So when you look at the base of the tree of life even now, it's not an accident that you'll see Thermatoga there, this thing we talked about today, which is probably just a name for you right now, but to get an idea is, Carl Stetter, the person they're talking about, the German scientist, was digging this stuff out of geysers. We start to see at the base of the tree of life a lot of hypothermophiles. You go like, well, why is that? Well, probably because these things evolved back when the Earth was a hypothermophilic place. So we have evidence, people that, as you saw, not only just isolated bacteria, they're talking about whole bacteria ecosystems. You can see people here getting samples, and I don't know what that guy's doing. He might be getting a sample and might be doing something else. So I think at this point we'll stop and we'll move ahead. The evidence now moves into things that we look back in hindsight at the archaea. We've talked about this one, for instance, okay, but can you still have hypothermic bacteria with these characteristics? Yeah, they have to have different adaptations, but we start to see some things that lead to divergence very early on and have big influences